So my name's Dan Wegmann. I'm visiting over from the University of Fribourg in Switzerland, and I'm going to tell you something about the use of ancient DNA. We're a bit struggling with the mic. Apparently there's a new speaker in the back, so I don't know, can you hear me? It's okay?
That's not working.
That's not working? Okay. I try to speak very loudly, okay? So, but before I'm going to go to ancient DNA specifically, I just want to kind of like briefly mention the general philosophy of what we're doing in the lab or what we're interested in. And that's generally, I think, what most of us do, that is trying to find a clever way to go from what I would call very noisy data to a biological insight. And so there's many different ways by which you can approach this. The one we are mostly taking, but we are very open about trying other things as well, is that we try to find a specific way or a way to model the noise explicitly. That is, we try to understand how the noise in our data, where it's coming from, and try to find an explicit model for that. We then try to embed that into a hierarchical modeling framework. And so the idea here being that if one data point is very noisy, we might be able to get some insight by aggregating information from multiple data points, and then be more interested in maybe hierarchical parameters rather than just a parameter associated with each data point, and then do some model-based inference. And in today's talk, I tried to kind of walk you through some of those ideas and how we apply them to the question of analyzing some ancient DNA. Before I do that, though, I would like to share with you very quickly one of my very first big data insights, and that traces back to about, I don't know, 15 to 16 or so years. So in Switzerland, it's actually compulsory to ski, right? So which sounds very weird, but it's true. So when I was in high school, like every school would run a mandatory ski camp every winter where you have to go and have to learn to ski. And so during my studies, I was working as a snowboard instructor in all those camps. And together with some friends, we were pulling together accident data. And I can tell you, Wednesday afternoons of 1 week courses are by far the most dangerous time for me, right? So I'm quite glad you're sitting, but just so you know, okay? Anyways, so we're gonna now jump to ancient DNA, probably more dangerous times, no snowboarding at the time though, but if we kind of go back in time to say 12,000 years ago, and I must warn you, this is a very computational biologist introduction for computational biologists about prehistory. So I don't know that much about archaeology, but if we go back 12,000 years ago in Eurasia, most people were actually hunter-gatherers. That means they had a foraging lifestyle and they were building or they were gathering their food in the surrounding. And then something quite remarkable happened about 11,000 years ago, and that is in what we call Fertile Crescent, that extends here also to the east. Kind of a new way of subsistence emerged, that is like people started to actually farm. And if you think about it, that's probably one of the most drastic cultural revolutions in our history, because along with just changing your food comes a lot of other things with it. So one is, for instance, people started to be sedentary. So you no longer followed large groups of animals for hunting, but actually they started to be or live in one single place, which also kind of culturally poses a lot of interesting questions. That is, suddenly people probably have to deal with property, right? Like as long as you're just following animals, I mean, it doesn't really matter whose square meter or square acre this part of land is, but if you're actually tending it, then you might actually claim this for yourself. So there's a lot of kind of changes that probably came along with this. So this is why I think it makes this period very interesting. From like the Fertile Crescent, farming spread relatively quickly to Europe. So it reached the European shores down here in the Aegean region, so Greece and Anatolia, around 8,500 years ago. And then the whole continent within a couple of next 1,000 years. Okay? And so, as we know, most of us, at least in Europe, most people are now living off of agriculture. There's interesting questions that come about like this kind of transition. One is, because this is just showing you a cultural spread, that shows like how the culture of farming spread, but the question is, was that mediated due to people, or actually just due to the idea of farming spreading itself? So one theory says, okay, maybe these were just hunter-gatherers seeing other people eating bread and saying, oh, that's so tasty, and then like switching their lifestyle. Or maybe at the other extreme is farmers just moving in and pretty much conquering the place and claiming the land for themselves. And of course, we can imagine all sorts of variations or things in between those two extremes happening. So when we started this project, there was a bunch of ancient samples analyzed from within Western Europe. These were both hunter-gatherers and early farmers, and some admixture analysis on their genetics. revealed an interesting pattern. So first of all, it seems like the early Neolithic, so the early farmers moving in, were genetically relatively distinct from the hunter-gatherers in the same place, suggesting that this kind of spread of farming into Europe was actually mediated by people moving in. But what we also see is that in later times, so this is kind of chronologically, in later times there was some admixture with hunter-gatherer ancestry reappearing in a kind of the farming population. So it kind of seems to be that between those, there's something in between those extremes that was happening. But what's still pretty much unknown is whether this kind of ancestry that we see in the early farmers actually really traces back to the Fertile Crescent, or whether there was something happening on the way. Okay? And so collaborators with us, they started to sequence whole genomes of ancient samples, both here in the Aegean region, so in Greece and Anatolia on the other side, but also from, so this point is a little bit misplaced, it's actually in eastern Iran, where we have one of the earliest farmer that I think was ever sequenced. Okay? And so we wanted to see whether this early farmer ancestry that we found here in Europe actually traces back all along the way of the expansion route or not. Okay? So this was the question we were interested in. However, like the reason why we got involved in the project is not because we know anything about ancient DNA, we actually didn't, but what kind of like made us interested in it is the fact that ancient DNA comes along with a lot of issues. There's lots of sources of errors and noise that contribute to ancient, or that are present in ancient DNA, and we felt that properly taking those into account was actually an important step in order to establish a relationship between individuals, right? So for instance, if ancient DNA is way more noisy, has way more errors, and we don't take that into account, then ancient genomes may appear to be way more distant from modern genomes than they actually are, because some of these errors will be maybe interpreted as real differences. Okay? So what are the special kind of like, or what is peculiar about ancient DNA? So first, the issue is that most of the samples we're getting have super low DNA content. So these are some of the samples from Greece that we analyzed, or that our collaborators analyzed in the lab. And what is shown here is the percentage of endogenous DNA. So how much of the DNA you get out of it is actually of human origin, right? And so our best sample is about 60%, so you're going to sequence 40% microbes anyways, okay? And then the other problem is that because the DNA content is so low, if you have to do PCR in the lab, and so in the end you don't get out that many unique transcripts. So even if maybe 60% of what you sequence is human, it can still mean that you actually resequence the very same fragment over and over again. So what is shown here is the unique fragments per library that we get. And what's a bit frightening is, so these dots here are controls that are seemingly containing no DNA, right? So there you get about this, and this preliminary run, about 100,000 DNA fragments out of it, or there should be none. And so our burst sample is on the order of 10 to the 7. So you get 100 times more DNA out of the burst sample as in essentially pure water, right? So this is kind of scary, right? So there's very little DNA in those bones, actually. The second thing is that most of the fragments you're getting out are super short. That's because the DNA was fragmented over time. This is shown here in the distribution of fragment lengths that we get from sequencing. So most of our samples have fragments in the order of between 40 and 60 base pairs, and not much longer than that. It meant that after a couple of individuals, we actually stopped using paired-end sequencing because the two pairs would just entirely overlap all the time, right? So these are the other samples that we got where we just did single-end sequencing, and it's totally sufficient for most of this, okay? So apart from low DNA content, we have those super short fragments. And then there's another interesting feature about ancient DNA that's called postmortem damage. So over time, there is cytosines that are actually deaminating in the DNA, and that results into C to T mutations that we see in the data. And so, or G to A on the opposite strand. If we don't take that into account and actually just use regular genotype calling on this, then we might see a lot of differences, a lot of mutations or variations appearing that have actually nothing to do with the variations that the person carried while still alive, but just got incorporated in the DNA over time. And so definitely we have to take that into account. One interesting feature about PMD though is that it seems to happen much more often or much more frequently when DNA strands are single strand. So that happens when you have a fragmentation of the DNA, then you lose nucleotides at one end, and the other end seems to be single-stranded, and that's where the chances of such a deamination is much higher. As a result, we find that the T to C kind of transition rate, if you just compare where you find a C in one of the reads, where you have a T, where you have a T in the read, while you have a C in the reference, is actually like almost exponentially distributed along the read. Of course, like there's constantly some difference because the reference is distant from our ancient sample, but there is a heavy increase of this at the beginning and ends of the reads, and that's also a way by which you can actually take that into account. Okay, so these are the three major issues we're dealing with. And so the goal was kind of to come up with a way to estimate genetic diversity and eventually maybe do some genotype calling even on these ancient samples. So given that they have very low sequence index, usually super short fragments, and have postmortem damage present. So if you have low sequencing depths and high error rates, then that kind of automatically translates into ambiguous genotyping. Okay? And of course, like, it seems like a super stupid idea to then actually do genotype calling and then try to estimate diversity from this. Right? I think we're all aware of the fact that what usually is done in a kind of a more medical context maybe is you try to genotype call and then apply a lot of filters on that, so you only keep the variants you really believe in, but that heavily biases our estimates of genetic diversity. Right? We should do the exact same type of filtering also on actually the or on the sites that are monomorphic in order to have a fair comparison of which sites are actually polymorphic in a sample. Okay, so that doesn't sound like a very good idea, and so how can we actually therefore turn like this noisy data into insight? That is, if you want to see like what was the genetic diversity of people in ancient times, how do they relate to other people, and so forth. And as I said before, like the way we usually do this is that we first try to find a way to model error rates explicitly, and then we're going to embed that in a hierarchical kind of parameter inference framework such that we can integrate out the uncertainty of the genotypes in that case. So we never want to do genotype calling, we want to integrate out the genotype uncertainty, and we want to have a way to aggregate information across loci, because for every single loci we have no chance of being sure pretty much what the genotype was at that. Okay? And so here is kind of one way by which you can approach this in a population genetics way. So I'm going to first talk about how we try to infer just genetic diversity from that data. And I must say this is work that was mostly led by two people in the lab. So Athanasios Kouzatanos is a postdoc in the group, and Vivian Link, a PhD student who is actually attending this course. Okay. So what we put forward is like the following idea. So we said if we cannot actually estimate genotypes at every site, what we would like to do is we'd like to estimate the parameter of interest in a hierarchical fashion, which in our case is heterozygosity, or just the genetic diversity. If you have a single individual that has two chromosomes, then these two chromosomes, they're related by some sort of tree or genealogy, okay? And this genealogy is very simple because there's only a single topology. So the only thing that characterizes this kind of genealogy is actually the time to the most recent common ancestor, which we're going to call T here. So therefore, the expected, or the number of mutations you expect to see in that tree is given by 2 times the time to the most common ancestor, which is just the total length of this tree, times the mutation rate. Okay? We will never be able to distinguish these parameters, but we can just call something zeta, which is the product of these two, which is the kind of the rate of mutations we expect to see in such a setting. And then another thing we definitely don't want to do is we don't want to use any reference information, right? If you want to check whether ancient people were very similar to modern humans, then if you kind of use modern knowledge, say on the reference genome or the variants present, and use that to infer diversity of ancients, you make them look more modern than they might actually be. So one idea is here is we really want to find a way to infer genetic diversity without any information that comes from a reference genome. And so we say that, okay, apart from like this simple assumption that we're interested in this parameter theta, we actually also want to have a prior on the genotype frequencies that is given by the local GC content, or like the frequency of nucleotides in the region. So if you're in a GC-rich region, you're more likely to have a GG, CC, or GC kind of genotype than another one. We want to use that type of information because we can learn this directly from that individual as well. With these kind of two things, we can build up a very simple likelihood model. That goes like this. So we're interested in the probability of all the sequencing data given the zeta, which is a parameter of interest, and the pi, which are these frequencies here. And so we want to try to find a way to estimate that. And we're going to assume, first of all, that if you go across loci, then all loci are independent. So we can do the product because mutations should happen independently between loci, or that's the assumption of the model. And then we're interested in integrating out the genotypes. So we have a sum here over all possible genotypes. We have the probability of the sequencing data given that particular genotype with 2 nucleotides K and L, weighted by the probability of seeing that particular genotype given the diversity of that sample in that region and the nucleotide frequencies in the reference genome. And this is how we want to estimate both zeta as well as pi. Okay. So it's relatively simple. We're just interested in now specifying these 2 individual terms, and then from there we can go and do some inference. So the first one would be how to get the probability of a particular genotype given zeta and pi, and then the second term we're going to look at is how to specify the probability of the sequencing data, a condition on the underlying genotype of an individual, while explicitly modeling the particular areas of ancient DNA. So this is the substitution model we're employing. It's Felsenstein's substitution model from 1981. It's quite commonly used in phylogenetics. It's a pretty standard model, but the neat thing for us is that it's actually including these two parameters. So first of all, the rate at which there is kind of a mutation appearing, and the second one is the frequency of the nucleotides at both ends. So if you're not familiar with it, don't bother too much about this equation. What it simply just says is if you're starting at one end of the tree and you're following the tree, so what's the probability that you start with an A and then there's mutations happening or not, and you end up with again an A, because there could be mutations going back to A, or a C or a T or something like that at the other end. Okay? And this is just it, and there's two expressions, one for the case in which you have the same nucleotide at both ends, and one where they have are different, because the second one does exclude the option of there no mutation kind of happening, right? Because if you have a different nucleotide, at least one mutation must have happened. Okay? But it's a pretty standard model, actually. When it comes to the what we call emission probability, so the probability of the data given the parameters of the model here, so we're going to formulate this like this, and I think it's easiest. So we have this problem of postmortem damage that turns C's into T's and G's into A's, which depend on where in the fragment you're actually placed. So what we see is, I guess it's easier to explain that with a little example. So the question, for instance, is what's the probability that in a random read at what locus we're going to see an A, given that the genotype is a T, and given that we know the error rate that comes from the machine estimate of the error rate of that position, a particular location in the fragment, in the read. And here a D, which is kind of a generalized postmortem damage probability that I'm going to go into a little later. So because postmortem damage has actually never turned a T into an A, the only way by which you can see an A if the underlying kind of thing you're sampling from is a T is that you're going to make an error, and we just assume here that an error turns into any of the three other nucleotides at the same rate. But if you have, for instance, if you observe a T and the genotype, underlying genotype has C, then of course there could be postmortem damage, so you have to specify that out. It could either be postmortem damage and no error, or it could be no postmortem damage and error, so you have to separate those. Okay? The question is, how do we get these postmortem damage probabilities? So you remember these shapes that is kind of like an exponential decay coming along the fragments. So we can actually learn that shape or this function first from all the fragments. So this is what we do. We fit the model to that. That takes also into account divergence between the reference genome and that sample. It's also something we estimate, but then we get what we call these generalized probabilities that we then can plug in, which are given after that step. Okay? Well, and then basically we just assume that each and every read is independent at the same site. That's a common assumption in genotype. Okay. Well, and I think from this we're done. So the first thing is we then use an EM algorithm, in this case, pretty straightforward, kind of implement an EM algorithm in order to get estimates. So one thing we're interested in then is like what type of sequencing depths would be required in order to get an accurate estimate of diversity of an ancient sample. So what I show you here is some simulation results where we have on the x-axis different sequencing depths for these different individuals, going from 0.2, a coverage of 0.2 or sequencing depths of 0.2 to about 3.2. And we estimate here the zeta, so these are replicates across windows of 1 megabase in size. So we have 1 megabase of data, and in that megabase you have a zeta of 10 to the minus 3, which corresponds to kind of like the average human diversity. And then we were wondering whether or not we can estimate that, and it seems there's a lot of noise if coverage is super low, but as soon as coverage is on the order of say 1x or something, then we start to like really zoom in on the accurate zeta, which is quite impressive. If you think about an average, you have a single read per locus, which is definitely not sufficient to call a genotype, but across 1 million sites is actually sufficient to say, have an accurate estimate of the diversity of the sample. Okay. What's also very interesting is that the same relationship also holds for window size. So if coverage is very low, what you can do is you can just increase the window size and then get an accurate estimate as well, which is really showing the benefit of aggregating information across the different replicates, right? So we can kind of compensate for the fact that our data is super noisy at one point by just using more points to get our estimate. And that's also shown here where we estimate the relative error as a function of either sequencing depths or a combination of sequencing depths and window size. And this is a density estimation of like our results, but it shows kind of like these two things perfectly compensate each other and are nicely here on the diagram. Okay, so point is, if sequencing depth is super low, we can make up for that issue somehow by increasing the number of loci or the window within which we want to estimate zeta. So that sounds all kind of super promising, and what we then also, of course, looked at is how the actual value of diversity affects these things. And all the data I showed you before was for 10 to the minus theta of 10 to the minus 3, human average. Of course, if we have much lower diversity, it's much harder to infer anything. Somehow, like within a window, the only way by which you can build evidence for there being a polymorphic site is at sites that are covered at least twice, right? If your coverage is low, few of those sites, definitely less than sites in the window. And the problem there is if the theta is 10 to the minus 4, which is one out of 10,000 sites, you need probably to have a couple of 10,000 sites covered twice in order to see that. And that's kind of what we see. Okay, so there's lots of noise related to that. But as soon as the true theta is higher, you're going to be more accurate estimates. But that's something to take into account, right? There is a relationship between the value you want to estimate and the error you're going to make. So that's kind of a little bit dangerous, but we have to keep that in mind. As soon as coverage is really above 1, right, so it's 1.6 or it's 3.2, then suddenly this looks much better and we're going to get pretty accurate estimates. Okay, so we were super excited, and then we said, well, okay, let's apply this to our Greek samples, and so this is kind of what we got. So this is just a part of chromosome 1, and 1 megabase windows. Each dot here is an estimate that's just connected by lines of 1 with genome, color corresponding to the different samples that we have. Okay. And what we see is, well, first this blue sample here is super diverse. It has, I don't know, this is 10 to the minus 1, right? It means 1 out of 10 bases is polymorphic. I don't know what that's, a little lower, but say it's 1 out of 20. That's kind of crazy. And then you have this guy here is around 10 to the minus 3, which you would expect maybe for humans. And this one so shows this super weird pattern between 10 to the minus 5 and 10 to the minus, I don't know what number, which is essentially 0. Okay. And so you can, I mean, the window is 1 megabase, right? If you get an estimate of 10 to the minus 10, it just means the EM converges onto 0. And so, well, now the question is this biologically reasonable? Like, does this make any sense? And I would argue it makes pretty much no sense, right? Like, you don't have any human that is polymorphic at every 20th position, or someone that has no single kind of polymorphic site along the whole genome. So, kind of what's going on, right? I mean, in simulations, this model looks very nice, but then somehow it breaks down when we apply it to data. And I think that for us, this was a kind of an important lesson that I would also like to share, and that is in all this modeling, we try to incorporate error rates explicitly, we put all these things in, but of course, if the error rates that we're going to use are completely off, I mean, then the whole thing should be off, right? So what that just tells us is, if I just may switch back slides to the equation, so what we do is, here in the emission probabilities, we kind of assume that we know the error rates at every single location, because that's an estimate we get from the sequencing machine. But if that error rate is, of course, wrong, so is our diversities. That's working with low coverage or low sequencing depth data. It means you really rely, or you have to trust the error rate given which is nucleotide. You cannot compensate bad, kind of badly calibrated base quality scores with more coverage. Okay? And we believe that this would actually explain the big issue that we got, that these differences are not due to biology, but due to kind of different ways by which base quality scores of these samples were distorted. Okay, so now how are you going to actually recalibrate those scores if they're so important? The classic way is you run BQSR, right, which is you take all the sites you know are known are polymorphic in modern humans, and then you mask them, and every site that is polymorphic, that any error that you see in another site you think could actually be polymorphic, but it just felt like kind of weird to us that we would use all this information about modern Europeans and can use this to recalibrate our ancient samples, which may be very similar to modern Europeans, but may also be very different, and that's exactly the research questions we were after, right? So we definitely didn't want to use all that modern information to recalibrate in this case. So how could we move about that? And it turned out that we were kind of like lucky in that, in the following sense. So all these 3 Greek individuals were actually male. Now, if I say we were lucky that they were all males, that's not because males are more interesting than females in any ways to study, but they have one interesting feature in this particular case, and that is they're actually supposedly haploid on the X chromosome. Right? So even if we don't know their sequence on the X chromosome, if we see reads disagreeing on the nucleotide, one of the two is wrong, right? Or must be. So, and the point is we can now exploit that information in order to learn something about errors. We can just say, we don't know the sequence on the X chromosome, but if 2 reads at the same position have a disagree in the nucleotide, one of them is wrong. I don't know which one, but one of them is wrong. Okay? And so this was kind of the idea that we're doing here. I'm just going to say that by now we figured out that we don't need a lot of data, and there's also ways by which you can apply this to see ultra-conserved elements or mitochondrial DNA and stuff like that. So I think we are now at the position where we start to also be able to study actually females, which is very interesting. But at this point, like, it was just developed for these male samples. So this is how we kind of approach this. So the idea is really we assume that there is a single genotype for every, or a single kind of nucleotide that's a true nucleotide at every position along the X, but you don't know which one it is. So for each kind of this is the what we're interested in this probability here is the probability of all the sequencing data at one locus, given a kind of a vector of parameters beta that are going to tell us something about how these quality scores are actually distorted. Okay, and I can explain how we use that beta after we're going to go through this equation. So this is what we're interested in, and we're going to say, well, I have to integrate out the genotype because I don't know the genotype at that location X in the genome, so I'm going to just sum over all genotypes. But then all reads at that particular locus, they have to be coming from the exact same genotype. So then I just have the product across all reads of the probability of observing that particular nucleotide in my read, given the underlying haploid genotype, given the error rate, okay, epsilon, given the postmortem damage that might happen at that case, weighted by the probability of the genotype, okay, which we have a distribution of these nucleotides in the genome that we can use here as a bracket. Okay. So, and then this is what we build up. The question is now, how do we bring in like our beta here with the epsilon? I tried to simplify the notation here a little bit. So what we do is the following. We say there is some logistic regression or some logistic relationship between the error rate, which is bounded between 0 and 1, and two quantities. One is Q's, which are something we know about those things. So for instance, that the actual nucleotide we see, it is the error rate reported by the machine, it is the position within the read, all the things you usually have in BQSR, and we can have a lot more of those things. So there's, but it's bounded between 0 and 1. And the way by which these informations, Q here, are contributing to what we plug in the logistic regression here, is just a linear function. So we're going to say, well, I have some weights, beta 0 is an intercept, and then I'm going to add like an effect for the position within the read. So maybe along the read things get worse or better, I don't know. We can plug in, so what we do is then we plug in the square of the position too, so that we have some nonlinear effects. We plug in the actual nucleotide at that position, we plug in the complex, so what was the nucleotide just before, and so you can build essentially your own very complicated model if you want, that would relate features that you know about one particular nucleotide to the underlying error. And then the goal is to find a way to estimate these weights beta from the data using a segment of the genome where we believe that there should be, every single site should be haploid. Okay? So this is the point. It turns out that you can again do that with an EM algorithm, relatively simply in this case, and so this is how we do that. So we estimate all these coefficients beta for a series, for a vector of information that we have for every site. Okay? So again, we use some simulations to show that that actually works. So we distort quality scores heavily with different kind of, by using exactly this model, of course, with different betas, and we try to estimate this beta as a function of sequencing depth. And so we found is this one here. So we found again, like if sequencing depth increases, we can estimate these distortions relatively accurate. Okay, this is true for, this is the position effect, this is, this is the quality, original quality effect, the quality squared, the position squared, and then there's lots of complex and other things that I don't show, but they all show pretty much the same pattern. Okay. What we also found again is the same, like you can actually just increase the number of sites you have if coverage is low. So if you have very low coverage, we can use just larger windows, like say whole X chromosome or something like that, which is way more than 1 megabase, which is used here to get accurate recalibration. Okay. So we then applied this to our 3 Greek samples, and so these are the quality distributions that we get. So we have here on the x-axis the quality score reported by the machine, and this the recalibrated quality score that we get out of once we fitted the model and then recalibrate each quality scores. To me, there's a lesson to be learned in that, like, this all sequence on Illumina, so Illumina seems to be super overconfident about its own quality, because somehow if it was on the diagonal here, it means the truly, or the reported quality is similar to the true quality score, but all our true ones are actually below the diagonal. So that suggests that there is way more error in there that the machine itself thinks. That's consistent across all the samples we have, which were all sequenced in multi-independent runs, different libraries, and so forth, and it's just overall true, right? So it seems that Illumina machines are a little overconfident about their errors. But we can recalibrate those, and if we do that, and now we're going to cheat a little bit, but if we do that for this and for other samples for which you're going to show results, we're going to get results like this. So we can now estimate heterozygosity or diversity along the genome here, and that's shown for different individuals. There's 2 1,000 genomes individuals, modern Europeans, and 2 hunter-gatherers, because these are the ones that are the most different, so this is why I showed these two. And so what you see or may not see, I don't know how easy it is to see, but you should maybe see that the blue and purple line follows each other actually quite accurately, which is also interesting. So it's not only that the two modern European samples we have, they're like generally similar in like their genetic composition, but of course if we just go along the genome and see how diverse individuals are in different regions, that itself is very similar between individuals too. We can get for each and every window, we have an estimate of diversity, so we can plot these, make these kind of box plots in which we plot the diversity of the whole genome in one leg of these windows for each of these individuals. See, these are three kind of modern individuals from Italy, from Tuscany. These are three modern individuals from Great Britain. This is a Bronze Age individual that we have, and that's very interesting. So the Bronze Age individual seems to have almost exactly the same diversity as modern day Europeans. In contrast, though, the two hunter-gatherers for which we have such data, they have a quite reduced diversity, which goes in line with kind of the assumption that they lived in much smaller kind of groups and had a reduced effective population size as a result of this. We also looked at some African populations, so these are three modern African Yoruba populations. This is the only kind of whole genome ancient African sample existing, Monta, and we see that the Africans have unexpectedly a higher diversity than Europeans, but interestingly, even the ancient African here, several thousand years old, had already a higher diversity than any of the modern Europeans have today, right? It's kind of like this pattern as in ancient time. Now you would say, wow, that's crazy, right? You read all these papers with like 200 ancient samples analyzed, and now here I put like 4. So the reason is that if you look carefully, most of these ancient samples processed, they have an ascertained kind of set of SNPs for which they use capture to go after them. And that tells you a lot of things, but it definitely doesn't give you an unbiased estimate of diversity. So the number of genomes for which you can actually do that analysis is unfortunately very limited. Okay? But I guess in the future you're going to see way more studies that are trying to produce actual genomes rather than just SNP data from an ascertained kind of SNP, or at least I hope. What's also kind of interesting, what we did here is we calculated correlations between the distribution of diversity in the genome. And what we find is, so these are all the modern samples, they cluster together quite nicely, so they're highly correlated. Based on this, we have the Bronze Age sample, which is also quite similar in distribution of diversity to the modern ones. The African seems to be very similar, based on this, the ancient African. But then the two hunter-gatherers we have, they are quite far away, quite distant from the moderns, but interestingly, they were also very different among themselves, right? So that is something other people claimed as well, so that the hunter-gatherers we have in Eurasia, they were actually very distinct or genetically quite dissimilar from each other, at least if you compare that to modern samples. I should maybe say that like this is maybe an unfair comparison. So this one is a sample from a cave in Switzerland, while this one is one from the Caucasus region, so it's also geographically, these two samples were of course pretty far away, right? Okay, we then also use the very same model of like errors and postmortem damage and so forth to develop the genotype caller for these samples. Of course, if the coverage or sequencing depth is very low, as I said, it's maybe not a very good idea to actually call genotypes, but sometimes you might be interested in, especially if you want to do PCA analysis. Well, you can work off the band files, but you can also work off like genotypes and try to do that in a clever way. But there's also other applications where it's interesting to at least have maybe genotype likelihoods, if not actual calls. And so we implemented that, and this is just a comparison between our approach, which we call Atlas, for analysis tools for low coverage and ancient samples. And we compare that to the standard GATK approach, where we also used for those in the field map damage, which is a way to cut off to correct the quality scores of potentially the sites that are potentially affected by postmortem damage, right? So this is kind of a way you can just downgrade the quality scores at potential PMD sites and then use the standard tool where we did that here. Okay, so there's maybe two things to notice. So the first one is that GATK, that's been shown before, but GATK is heavily reference biased, right? Like you see that here in the following way. So the solid line is actually the fraction of bases at which the caller makes an error. Okay, so we used here simulated data, and this is the fraction of sites at which the caller does not emit a call. Okay. And if you compare the blue line, you see that the error rates we make is as a function of sequencing depths. Okay, these are the errors, this is the rate of errors we're making in reference genotypes. This is alternative genome, and these curves are identical, right? Because Atlas, we don't have any reference bias. We definitely don't want to bias towards a modern sample. So the error rate in the calling is exactly the same between these two genotypes. It's a little higher here for low depths in a heterozygous case, of course, because you just call them as a homozygous one if you don't have sufficient data. But then if you looked at GATK, so GATK has a much lower, a significantly lower error rate in the reference call, but actually quite some higher error rate in the alternative call. Okay? In addition to that, GATK just does not emit a lot of the alternative calls. So like here it's up to more than this is 10%, right? So if you have a sequencing depth of 2 or 1 or 2x, then at these sites, GATK does simply not make a call at more than 10% of the sites, if it's alternative. But if it's reference, like this is much lower. So which means if you factor these two things in and just call with GATK and then compare, you have heavily biased towards like reference call. You're going to have way too many homozygous reference calls and way too few homozygous alternative calls. Okay? And so our caller does not have this this kind of bias, and it also emits we are kind of like make hardly any kind of decision nodes to emit something. We actually, personally, I really believe that if you have the proper genotype likelihoods, like you can always report them, there's no reason not to, right? They can be very similar among the different genotypes, but that's still information, and so we output it. Okay? So in summary, it seems it's quite easy to beat GATK when you were looking for unbiased kind of like genotype calls, and if we incorporate postmortem damage directly into the model, then we also have a much higher accuracy than GATK. Okay, so let's go back to biology, like the original question that I said, like let's see where kind of these farmers were coming from. I'm just going to show here a very, very simple type of analysis. We did more complicated analysis, but this is the graphically most appealing. So what I show you here is a PCA plot spanned with modern data. So these are all modern Europeans. We have here Slavic populations from Britain, Central Europeans, Southern Europeans, Sardinians being the outlier, Near Eastern population, Western Asian and Caucasus populations. And now we want to know if we project our ancient samples onto that plot, where do they fall? So first we use existing samples for both European foragers from actually within Europe, as well as early farmers from within Europe. And this is the same data that was used earlier to show this distinction. So this is captured data. And what's interesting about it here is that there seems to be a huge difference between these two groups. And this was also this kind of result. It's not ours, this is not our data. It was shown before that actually the early farmers were genetically quite distinct from the hunter-gatherers within Europe. But the question now is, where do the kind of individuals fall that we got from the Aegean region as well as from the Fertile Crescent? So first, if we project our first Aegean farmers, so these were the earliest farmers arriving on the continent. Right? If you think on a temporal scale, there's no farmers in Europe. The first ones stepping on the shore arrive in the Aegean. We get samples from precisely those, right, just a couple of 100 years after they arrived. And then these red guys are like 2,000-3,000 years later, the first farmer in Europe. And they are falling on the same place in the PCA spot, suggesting that they were genetically very similar. In that case, that actually, I think we can say that the early Aegean farmers were kind of the source of the demic spread of farmers into Europe. Right? That doesn't make the whole chain to the Fertile Crescent, but we know at least that the first farmers that arrived on European shores, these were the people, or these were that actually then later colonized the rest of Europe and mixed with hunter-gatherers. Okay. So where does the Iranian farmer fall? That's also interesting. So our Fertile Crescent individual is kind of up here in a totally different position, suggesting that actually the first farmers in the Fertile Crescent itself were genetically relatively distinct from the first farmers in Anatolia and the Aegean. So suggesting that at the very onset of farming, things are a little bit more complicated. It's not just one group of people that developed farming and then expanded from there, but it was probably a mixture of people that did that, either independently or there was maybe some cultural diffusion, and then you have later people picking up, right? There's since been another study just coming out this year where they looked at this a little bit more detail, and they have this very complicated plot of who's related with which one, so things turn complicated. But I think what even this shows here is clearly there is a direct connection, but there is an ancestry relatedness connection of early farmers within Europe, but this kind of ancestry does not trace back all the way to the Fertile Crescent. Something happened there that's a little bit more interesting. Okay, so with that, I'm just going to come to the conclusions here from a maybe more methodological point of view. So the first one I think is that errors are kind of unavoidable part of working with NGS data, and I think we should deal with it. And I think as statisticians, computation biologists, bioinformaticians, filtering is really not what we want to do. Kind of like if we just say like, oh, I don't trust this thing, we can introduce a lot of biases in our analysis. I think if you're just looking for a GWAS hit, then it's maybe the right thing to do. But if you're interested in kind of the genetic diversity of some people and you just start to throw out random things, that's definitely not going to give you a non-biased view. So I think generally we should move away from using filters to decide what we trust, but actually rather try to incorporate the uncertainty that we have properly in our model to the personal state. Okay, so what I think for doing diversity of population genetics, what we should try to do is that we should also try to avoid actual calling genotypes, so that we have to make a decision, because we're going to be very uncertain about that, especially if you have low coverage data. But instead, maybe use hierarchical models that can aggregate information across different sites. So we're not interested in the genotype at one site, but some biological features from many sites. And that also allows us to actually integrate out the uncertainty at one site when we build confidence in that estimate. If you do that, there's another thing, kind of interesting side effect, which we are really interested in. That is, if you use these type of methods, it means also we can invest our budget in biological replicates rather than technical replicates. What I mean by that is we can maybe think of a GWAS framework. Okay, if you have to be very sure about the genotypes, you're going to spend a lot of money sequencing each individual. And if you have a limited budget, that just means you're going to have less individuals you can sequence. But if you instead say, well, how about we're going to have very noisy data for each individual? So I'm going to say I'm totally happy with having uncertain genotypes because I account for that in my method. Then it means I can lower sequencing depths of each individual, and for the same fixed budget, I can just have more samples. And that's what I mean by kind of shifting our budget from technical replicates, which is just sequencing the same guy over and over, to like kind of biological replicates. And I think that's interesting. Maybe there's enough kind of money around for GWAS data, but if most people studying no-model organism, that's a real problem. But of course, like there's one caveat, as we saw, if we are relying on estimates of error rates for each base to incorporate that in the models, they have to be accurate. And so base quality recalibration seems to be a very important step of this. Okay, and then I just want to do some PR as well. So all these kind of tools and a lot of other things that we develop for HDNA are kind of coded in a program that we call Atlas. So these analysis tools for low depths and ancient samples. I should really say that Vivian, the student here, is very much the lead author of this package. And it's a series of tools for HDNA that properly account for postmortem damage throughout everything that you can do, from really kind of like base quality recalibration, genotype calling, estimating a lot of things. It produces Beagle files for downstream analysis and so forth. Okay? It is also completely reference-free, which we feel is very important. Okay? So I'm going to end by just thanking a lot of people. So with all these, even though we just analyzed pretty much 4 genomes, right? You have a huge number of people involved because it's a major endeavor. So these are the major people in my group that contributed to this, Athanasios Chrysotenos and Vivian Link. And then this is a very close collaborator of mine, a mathematician, Chris Leuberger. He really hates having his picture in talks, but he's totally fine with this, so I mean, I put it. And these are the people that actually generated the data in the lab from our collaborators in Mainz, and of course there's lots of people that helped in the analysis, and all the archaeologists that were involved in this study, either digging up bones or just giving or helping us in the archaeological interpretation. Okay. Well, thanks all.